From Microbe TV, this is Office Hours for Wednesday, October 4, 2023. I'm Vincent Racaniello, and welcome to the corner of the internet where you can talk about viruses for an hour or two every week. I'm really happy to be here and welcome all of you. I want to thank our intrepid moderators. Who do we have tonight? We have Steph SF. We have Tom Steinberg. We have Les Fabi. Thank you all for joining us tonight. Small crowd tonight. Maybe it'll get bigger. It's no problem. Anything greater than one works for me, folks. What do we have going tonight? Let's see who's here. Let's say hello to folks. Tom, of course, is in Madison tonight. Here we go. And we know Les is from California. So is Steph, which is also from California. Neva is from Buda, Texas. Wonderful place. I've never been there, but I hear it's wonderful. Will is from Changsha, China. John is from Minneapolis, Minnesota. I just pick out where people are from, and in the meantime, the numbers go up. It makes me feel really good. Kang is from Chicago, 26C. Look at MK's in Theater, Greece tonight. Must be on location, right? Excellent. Enjoy. Not going to be here. Frank is from Santa Barbara, California. Ooh, 81 degrees. What is it here? It is... It's 21C. It's about 68F. Pete is double jabbed from London. He has 2 AstraZenecas, 2 Pfizers, and 1 Moderna, a full house, and flu, of course. You know, FU is not a good thing to say here, so we're gonna get past that. Lori's in Tomball, Texas, where it's raining. That's good. Elena is in Westchester. Elena, you got to come visit the incubator one of these days, right? Noir is from New Mexico, where it's probably very nice weather. Claire is from the UK. Joseph is Ontario. Questella is in Ohio. I saw you on a very, very old, one of our programs, really old, years ago, and you had said hi, so I said hello. Salinas Valley, California. Boulder, Colorado. Susan's from Boston. Love the Amy papers. The Amy papers are still up. They're very nice. Yeah. Really, really good stuff. San Diego. New Hampshire coast. Ooh, there's so many cool places here. Santa Rosa. Saudi Arabia. Pueblo, Colorado, Brazil. Oh, look at this, Norway. We have a lot of people from across because of the time differences, so appreciate it. Rach is from the Isle of Man. Karthik is from Charlotte, North Carolina. Thanks for all the episodes lately. Good to see you every week for 3 years. Yeah, I'm here every week. Try the veal. Thank you, Linda, for your contribution to science communication. Thanks for answering my questions the last couple of weeks. I'm over COVID. You've taught me a lot from Las Vegas. I actually made a mistake last week. You know, I said you could, after you recovered in a couple of days, as soon as you felt better, you could get a vaccine, right? No, it's wrong. I asked Daniel, and he said 3 months. It's the same. That's what the CDC recommendation has been all along, 3 months. So 3 months, if you've got COVID, 3 months and they get vaccinated. Why is that? Because the lymphocytes are recognizing the antigens from that infection, right? And you want to let them settle down before you give them a boost so they don't get too distracted and don't finish their tasks, right? We're off to Lanzarote next Wednesday. We'll miss 2 office hours. We don't have office hours for the next 2 weeks because I'll be in Boston next week and in Chicago the following week. So you'll have to learn how to do without it, unfortunately. Westerly, Rhode Island. Karen, hello. Michigan, Detroit Metro. Elicio, it's 33 hot and dry. I think it's from Brazil. Yep. Single jabbed from Wisconsin. And Kip and Laura from San Francisco, thank you for your support of science communication. Yes, flu jab. That's not the other one. We don't want to say that. Los Angeles, 30 degrees C. Owego, New York. Hello, Lisa. White Plains, New York. New York, Cohen. Asheville. Airy ozone. Asheville, North Carolina. Crestville Hills, Kentucky. Seattle. I want to say hello to Konami. Wayne from Urbana, Illinois. Thanks for pronouncing my name right the first time. Sometimes I get it. I don't always get it. Italy, Nicola. Hello, Nicola. How are you doing over there in Italy? No office hours for 2 weeks. No! You'll get over it. You will get over it. Don't worry. And then maybe you won't need it anymore. Who knows? You know, whenever you don't do something for a while, people leave. What can you do? Albany, New York. Rushby. Thank you. Is that how you say it? Rushby? And just in the house, it's going to pop up here in a moment. There's a little delay between the actual stream and when it shows up here. Here we go. Vanity Nutrition, thank you for moderating tonight. Andrew is going to sit this one out. Has to go to town for a B12 jab. Okay, good luck with that B12 jab. James is from Longmont, Colorado. Jan is from Vancouver Island. Okay. I love the crowd always. We have 113 people, which always exceeds my expectations. I never know who's coming. And there are many people missing, right? But that's fine. Rochester, New York. 2 a.m. in Italy, but still wide awake. I think there are people all over the world that need sleep, and they're staying up. But I appreciate that. And if you fall asleep, hey, maybe I can do ASMR. Put you all to sleep. Put you all to sleep. You guys know what ASMR is, right? Okay, enough of that silliness. Okay, let's go up and see what's happening here. Before we get to questions, by the way, I wanted to, you all know, of course, about the Nobel Prizes this week in physiology or medicine, right? Physiology and or medicine is always very exciting. So let's take a look. Look what we got here. This is you can go to the nobelprize.org website. And there's a nice little press release. And of course, the 2023 prize for physiology or medicine is given to Katalin Kariko. I went to a website to learn how to pronounce it. And Kariko, and Drew Weissman. And here's the announcement for their discoveries concerning nucleoside base modifications that enabled the development of effective mRNA vaccines against COVID-19. So not the vaccines. They're not getting the prize for the vaccine. They're getting the prize for the technology that they developed in order to have the vaccines made, a couple of years ago. Not for vaccines. As far as I know, there's only one Nobel Prize for a specific vaccine. Does anyone know what that is? I'm going to give you a minute to say, what is the one Nobel Prize that has been awarded? It's right there on the page. Here you go. In 1951, Max Tyler was awarded the Nobel Prize for developing the yellow fever vaccine. So that is the only actual vaccine Nobel Prize, okay? This one, Kariko and Weissman, is for the technology that enabled the making of the vaccines. And that's really cool. I think that is, yes, Tom guessed yellow fever. You got it, Tom. This technology is cool. So people for a while wanted to use mRNA for vaccination, right? mRNA encodes a protein. So how do you make vaccines? You can take a virus and inactivate it and inject that into people. Max Tyler made an attenuated vaccine where you somehow passage the virus. He passaged it many times until it lost its virulence. But wouldn't it be nice just to put one protein in? So to do that, you could put an mRNA vaccine in, or just put mRNA in. The problem is, and all this is summarized in this nice article, when you put mRNA into cells, you get inflammatory reactions. In other words, the host and the cells within the host recognize the mRNA as something foreign, and they mount a reaction to get rid of it. And so that limits, right, the mRNA's ability to make protein, right? If it's degraded right away, you can't make any proteins. Tom got that answer from my virology course. It's not a bad course. So Katalin Kariko, she was trying to get around this, and she was at the University of Pennsylvania in the 90s, and she wasn't getting funded. She wasn't getting any respect. Nobody thought her idea would work. But she happened to run into Drew Weissman at the university, and they started collaborating. And, they noticed that, okay, first of all, dendritic cells recognize the RNA, that if you inject it into a mouse, as foreign. And so they said, well, why doesn't mRNA from a mammalian cell do the same thing? When you inject that, it doesn't cause an inflammatory reaction. It turned out that it was base modification. And they eventually found that if you modify the mRNA with pseudouridine, so one of the four bases is uridine, if you make it chemically slightly different, pseudouridine, it's not recognized and you don't get an inflammatory response. And so they began to test that by making vaccines against Zika virus and MERS coronavirus, and then they published these, and here are some of the nice publications. And we actually talked about one of these papers on TWIV 860, January 2022. Not too long ago. Almost 2 years. What am I talking about? TWIV reviews a seminal paper showing that chemically modified bases in RNA suppress recognition by toll-like receptor, a finding that was essential for the development of COVID mRNA vaccines, evading a toll on the road to RNA vaccines. So we have talked about that. I think this is really good. I think this is a good prize because the technology is very cool. It enables you to make many vaccines. And, of course, it was used to make COVID vaccines, which were really effective at saving many people's lives. And so it was a logical segue from them making it. They tested a few, and then suddenly you have this need, and so they developed. And of course, concomitantly at BioNTech, they developed their own mRNA technology, but using license from Carico. She licensed the technology to them. And so both Moderna and BioNTech were both using the technology developed by Carico and Weissman, and Moderna and Pfizer eventually market the vaccine. So I think it's a good Nobel Prize. I wouldn't have said it was good if you gave it for the vaccines themselves, right? But this is not for the vaccines. This is for the technology that enabled the vaccines. And so should someone else be involved? No, I think these are the two. I mean, these two push this forward. As far as I know, I could be wrong. If anyone has some information about that, tell me. I think this is the technology that pushed the vaccine forward. They did it, and they should get the prize. So I don't have any issue with that. Hat just said, does Dr. Offit know Dr. Carico? I don't know. He might. He certainly knows Drew Weissman. He's talked about him a few times. He probably knows Carico because she was at Penn for many years. Now, I'm sure you'll see in the coming days people claiming that they should be getting a Nobel Prize for the vaccine. They should have been included, right? There's a certain person who thinks that he was essential in this technology, but I don't see the evidence for that. So I think this is the right choice. The Nobel Committee does a lot of research. Sometimes they get it wrong. They get it wrong because the discovery turns out to be not such a noteworthy discovery. And maybe maybe the lipid nanocoating should be part of that too. I don't know. I don't know how much they innovated there. I know they made it so that it would be, immunogenic and come apart in the cell, and there was a lot of trickery involved there. And I don't know. So Susan Schmidler is saying, I read that the guy who developed the lipids delivery of the mRNA did not get any mention. Yeah, so that's a good question. That's a good question. All right, so that's thanks to Peter. Peter suggested that we chat about that. Oh, so Pete also said, with a vacant spot for the third nominee, is someone missing? I I don't think so. I think that this is it. I don't know about the lipid technology. I don't know how much that was already developed, and it went in little steps by a lot of different people, not just one person. I'm not familiar with that. So that's my thought. Thank you, Pete, for having that. Neva, viruses and microbiology are so endlessly fascinating. It's great to listen to this pod as an escape from the BS of U.S. world affairs. Well, some people don't like science. They just want to talk about politics, and that's fine. But there is a lot of BS, right? I mean, look at all the stuff that happened with the Speaker of the House this week. It's crazy. It's crazy. And by the way, there are other prizes too, besides Nobel Prize for. So Les tells me that they won the Breakthrough Prize this year. Oh, I guess you're talking about Carico and Weissman. Yeah, so they got that prize too. Let's look it up. The Breakthrough Prize. Yeah, that was money given by Sergey Brin, Priscilla Chan, Mark Zuckerberg. Winners of the 2023 Breakthrough Prize in Life Sciences. Okay, let's just get to it. Clifford Brangwyn and Anthony Hyman for discovering a new mechanism of cellular organization. AlphaFold is another one. Quantum information. I don't see anything telling me what it is that Les is talking about. So, Breakthrough Prize is another one. Okie dokie. So ask questions, folks. Yeah, there's the Pete one. Would it be consider starting off tonight with? Yeah, so that's what we did. Thank you very much. Did you come up with some questions, folks? They also won the Lasker Prize. Last year or so. Yeah, the Lasker is kind of like a predictor of Nobel Prizes, right? So Alan had tested positive for COVID, started Paxlovid a few hours later. 3 days of symptoms, sinus congestion, and sore throat. So glad I had my plan ready, thanks to Daniel and Vincent. You're welcome. I'll say you're welcome for Daniel, because he's the one who's driving that. And, you know, we get a lot of pushback all the time. But you should, you know, people who are dying typically don't get Paxlovid in time. WHO has decided that influenza B Yamagata is now extinct and should no longer be added to flu vaccines. That's an interesting question. I think this came up last week, if I'm not mistaken. I think you should wait. I'm pondering here, right? I want to give a quick answer. What's the rush? Don't be afraid of silence. Get your Twiv mug. They sell these at cafepress.com/Twiv. I think I have a thingy here. Nope, that's the Amy Papers. By the way, the Amy Papers are still a thing. That's very cool. You should check those out. Here, Cafe Press. Nope, that's Contribute. Well, anyway, cafepress.com/Twiv. You can get your Twiv mugs. I think they should wait. I know it's been some time since they've seen it, but you know, your ability to say that a particular virus is gone is only as good as your surveillance, right? So wait, don't rush, because maybe it's hiding somewhere. We haven't detected it. Then as soon as you stop vaccinating, you're going to have a lot of people who are susceptible, and then you'll have it back, roaring back. So I would wait a bit, okay? But they're not asking me, right? That's fine. You shouldn't have to ask me. We got that. Let's see, some questions down here. Is there any chance that the hosts of the Immune podcast could discuss the fairly abundant immune cell changes in long COVID? I think a lot of viewers... Yeah, that's the Iwasaki paper that we did on Twiv. I don't know, Susan, did you hear the Twiv? Because Brianne was on that episode. And although she didn't really weigh in on most of these changes, we could do that. Sure, I will pass that on. You say, well, okay, what does it mean that this cell population has gone up, right? That's what you want to know, yeah. Lori will be doing a lot of air travel over the next few months, so went ahead and got a COVID jab. Yeah. I don't think it's a problem. I think, as Paul often says, if you want it, just get it. So we recorded a nice article. Beyond the Noise with Paul Offit yesterday. It'll come out next week because we just released one this week, right? Anyway, it'll come out next week about this idea that why can't we discuss science when it comes to public health, right? If you say, I don't think this vaccine is doing what you think it does, why do people then call you names, right? Why do they say you're an anti-vaxxer because you're not all in favor of this latest booster? And Paul's very frustrated because he wants to talk about the science, but he feels that when it comes to public health, you're either in it or you're not. And he wants to talk about the science, right? So, yeah, anyway, that's a long way of saying it's fine if getting a jab will boost you. And this is matched to circulating strains. So you're going to get a 3-month boost of antibodies. It'll really dampen down. It'll prevent even a mild infection, most likely. So that's the idea, right? Probably work, but they couldn't test it before deploying it, of course. So you have to go a little bit on faith, and that's fine. Rach doesn't have the latest COVID jab here. So actually, in a way, it's an opportunity. I know you don't want this opportunity, but it's an opportunity to say, okay, we don't have anyone getting the XBB.1.5 vaccine on the Isle of Man. How do they do in the next season, this next COVID season, right? Now, I shouldn't say season because we don't know when the season is, but in this winter time when we're presumably going to get a lot of infections, how are they going to do not having this new vaccine? It's by no means a perfect study, right? Because it's not controlled. We don't have a group that's getting vaccinated, and we can't control for population differences, for age, for whether people had COVID or not, whether they were vaccinated before or not. But still, I'd like to see overall, maybe there's not much serious disease because the other vaccines were working. It would be nice to know that. So that's my thought, but it won't be done, right? I will get my COVID shot in 3 months. The shot did what it said, no hospital or serious illness. Okay, after getting infected, right? Took a bit to find tonight's live stream. Why? Just have to go to my YouTube channel. Just go to youtube.com/profvrr, and it should be right there. Sorry it took you so long. I'd like to ask Angela Mingarelli which cat viruses are truly zoonotic. So, zoonotic for a cat. So for people, it comes from animals to people, right? That's a zoonosis. What is it called when it goes from an animal to an animal? Even if it's a different species, right? So I'm not sure what you mean, Noir. Noir appreciated my ASMR, right? You can take this and go to sleep with it. This is what some people do. They rub their microphone, what do you call this thing? Windscreen. So Noir is with it. Truly zoonotic for a cat virus. So, yeah, it would have to come from some other animal, right? That's what you're talking about. Well, Angela's going to be on, where? When? Friday. Friday. So if I could get my act together, I don't have it. Let me write it down. My little piece of paper, which is going to get lost. Which cat viruses are truly zoonotic? And so thank you, Noir, for correcting me on how to contagious to people. Oh, contagious to people. Got it. She would know that, yeah. Yeah, for zoonotic from cats to people. Of course. What am I, dumb? Yeah. What cat viruses can affect people from cleaning and handling them? Yeah. Yeah, I will do that. Some people don't like the ASMR. Some people, if you search ASMR on YouTube, you're going to get hundreds of of videos of people rubbing their mic covers, clicking their fingernails together. Yeah, all kinds of stuff to make this noise that they say hits a certain part of your brain and you get that frisson in the back of your neck or sometimes puts you to sleep. Okie dokie, here we go. I wrote that down. Thank you, Nora. My mom is in the mid-80s. Not sure there's any point in her getting her 5th jab. So Paul Laffa would actually say, if you're over 65, you should get the jab. So that's what Paul says. I don't think it's a problem. You're at risk, and maybe if you prevent that mild illness, it's good at that age. John, thank you very much for your contribution. I think I already thanked you, but it doesn't hurt to thank again. All right, Ruby. Okay, it's like the gem, the red gem, Ruby. So funny. I love it. Ruby. You know, I was trying to be fancy. Rushby, right? Ruby. Okay, I hope I remember. That's the thing. In Michigan, lots of masks coming back, and doctors are starting to check in from car again. Pharmacies are full of people getting flu or COVID vaccines. Have to check our figures here in Oakland County. Yeah, the numbers are going to go up. It is perfectly okay to wear a mask, right? No problem there whatsoever. It is perfectly okay to get the new vaccine. Anything that you want to do that makes you feel protected is fine. I haven't seen masks in healthcare settings here in this area anyway, but of course it always depends where you are. I love wearing my spike protein shirt, but I worry that someone will ask me to explain what it is and that I won't be able to. That's very good. That reminds me, Nat. So what you could say is that this is a representation of the spike protein, which is part of the virus that causes COVID, and which that protein was what many people got in their vaccines, in their COVID vaccines. Yeah, it's a great picture. It's a great shirt. So on this shirt is a representation of the spike protein of the virus that causes COVID, and that spike protein is the basis for all of the vaccines. Yeah, that were used. Either you don't have to tell them this part, but the mRNAs encode spike. There were some protein-based vaccines which were pure spike, and then there were adenovirus vectored spike vaccines. Yeah. Okay, so Nat reminded me to show you a picture. What is it? Here. So this is made by Karen, my assistant, who got me to wear. This is one of my spike shirts. It's blue, and she hung a SARS-CoV-2 virus on my head. I have a mask here, and she hung a poliovirus on my shirt. And actually on the cape, there are also other viruses, which you'll see in a minute. Now, this is me. Yeah, I'm Earth's virology professor, so I'm overseeing the Earth. I'm teaching you guys virology, which is my honor, actually, to do that. I think it's great. Now, here is another one, which I really like. Let me make it a little smaller so we can see the whole thing, because the part that's cool is in the clouds. This is another one that Karen made. So she had me So here's the cape with the viruses hanging off of it. This is SARS-CoV-2 on my head, poliovirus on my neck. The black one at the bottom is HIV. The yellow is hepatitis A virus. And I think the blue is a rhinovirus. And this cape came from vaccinated.us. Where is vaccinated.us on my... Is it here? Nope. Is it... Nope. Vaccinated.us. I'll make one. Let's make a vaccinated.us text. You have to go there. Here we go, vaccinated.us, and you can buy. Unfortunately, the capes are out of stock, but you can buy spike shirts. And for Halloween, we're doing a promotion again with them, where they'll give the majority of the profits on everything sold to Microbe TV, and you just need to use the promo code Microbe TV on checkout, and you'll get the profits from the sale of that shirt will go to Microbe TV. Most of the profits, not all of it. They have to keep a little for themselves. So that's great. Matt from vaccinated.us, thank you so much for that. And this is a little promo I made. It's cool. She did a great job. Okay, let's turn it off and go back to questions. Thank you for that, Spike. So that's what Spike... I know it's not exactly on topic, but finally papers like the Post are starting to expose the damage sugar can do. As a healthcare worker who received one too many donuts as a thank you, I am overjoyed. Yeah. It is not exactly on topic, although sugars, of course, are part of proteins on biological membranes. The spike protein has sugars on it. But yeah, you should stay away from the sugary stuff. You know, a lot of people drink sodas all the time. No good. Cut back on the sugar. Yeah, you'll live longer. Last watch Tweevo live at 30,000 feet. Yeah, I guess you could do that. You get the onboard Wi-Fi, right? That's what Vanity must do. And yes, John is right. Amy would mention Kadiko and Weissman from Penn all the time. She liked their work, yeah. So HPV got a Nobel Prize for the discovery of the virus, not for the vaccine. And no, Salk did not get a Nobel Prize for his vaccine either. But you're guessing as to the only Nobel for a virus vaccine. And Tom got it right. Yellow fever, which he remembers from our course. A year ago, plus or minus a day, we visited the Nobel Museum in Stockholm. Info on the history and all the prizes and a few pieces of historic lab equipment. Yep, I visited there in 2019. And they had the dress that one of the women Nobel laureates wore. I can't remember her name. She bought a gown, a long dress for the ceremony and donated it to them. She never wore it again. I guess didn't want to wear it. But I guess, wear it to the ceremony. You donate it. That's cool. And there was a lot of other equipment. There were microscopes and there were cell culture dishes and so forth. It was pretty cool. Yeah. It's still in the Isle of Man. Always hard to get these abbreviations, right? Because IOM is in the U.S. Institute of Medicine. Anyway, in the Isle of Man, it's still the old vaccine. Yeah. Is there any point? So I don't know what you mean by old vaccine. If it's the original one, no. There's no point in getting it because it's so drifted now, and she's probably got enough T-cell immunity, so that wouldn't be worth it. But the new one, yes, I would say. Discussion on the last TWIV. It was said that there are no auto antibodies seen in long COVID, But Dr. Iwasaki stated that in a subset of females that they see autoantibodies against self. Well, in the comment that there are no autoantibodies involved comes from that paper. They found no correlation between the presence of antibodies and long COVID. Right? So in a subset of females, they see autoantibodies, but it may be that you also see that in a subset of females without long COVID, right? So that's even though you see it, it may not be meaningful. And in fact, in the big paper that we did, it's not meaningful whatsoever. There's no correlation between autoantigens and long COVID. Your virology course is a gift to humanity. Thank you so much. I really appreciate that. I thought it would be good for the world to have it. So that was my idea when I first started teaching the course in 2012. And it's been useful. Yeah, thank you very much. I like the way Vaccinated U.S. celebrates the COVID vaccine in their wording. It's very good. I read it. Remember, I read it on the last TWIV, and it's really well done. Yes. Getting a hooded sweatshirt. Excellent. Now that Amy can't be on live streams, any plan to have Dixon with you regularly? I love when Dixon just goes on tangents and tangents on TWIV. Seems perfect for a live stream. You know, that's not a bad idea, Nicola. I will ask Dixon. I just realized I could put my sticky on my laptop, and that way I will remember to bring it to the incubator. Okay, so this one, Dixon on office hours. Yeah, I think he would do that. He was on once, right? Yeah, that would be good. And Noir says, yep. Okay, we'll have it. Would also like to see Kathy. I've thought about Kathy. I've thought about Alan Dove. I've thought about Cindy Leifer. I could go through all the hosts, right? Yeah, I should do it. It's just a matter of doing it. Here's this comment from Susan. I read that the guy who developed the lipids delivery of the mRNA did not get any mention. So I'm just not sure how much. Yeah, I know that it was a big deal, but I'm not sure. Lindsay Marr got a MacArthur today for work observing how viruses move through the air and how much transmission occurs via the breath and fomites. Have you heard about her work? Let's see. I have not. Lindsay, American scientist. She's at Virginia Tech. Her work considers the interaction of nanomaterials and viruses with the atmosphere. Okay, that is a great idea. H-index. You know what the H-index is? It all about citations, I think. Okay, we'll get Lindsay on. I'm going to keep her page here. That could be interesting. And she got a MacArthur Award, so someone thinks she's doing good things. How do I find my H-index? The maximum... it's about an author-level metric that measures both productivity and citation impact. Okay, so let's search for mine. Vincent Racaniello. And the first thing that comes up is net worth. H, it's wrong, by the way. I'm not worth anything. what is my H index is 55, so Lindsay's is higher. I have 142 publications, and they've been cited 11,744 times, and 435 of those are highly influential. I don't know what that means, highly influential. My H index is 56. Yeah, one of the highest ones is a Nature paper where I was buried in the middle. It's okay. I don't mind. Okay. I can go riffing on this stuff. So Alicia says, I bet the next Nobel Prize will go to Raul on NOPV. I don't think so, Alicia. Well, first of all, he's modified an existing vaccine, so I don't think that's going to count. But also, his modifications got lost after the viruses went through people, and they still paralyze people. So I don't think so. What does H stand for? I don't know. What does H in H index stand for? It's one of those Hirsch index. HIRSCH. I don't know where I saw that. It must have been on the actual stream here. Yeah. MN asked that. Plasmid DNA residue in mRNA vaccine manufacture. I think it's just BS. I meant to look at this. I wanted to see. So, the mRNA is transcribed in vitro from a DNA template. So the question is, how do they get rid of the DNA, right? And my guess is that they digest it with DNase, but maybe not. So let's ask it, how is DNA removed from mRNA vaccine? Because I don't believe it's purified. The best way to go to this would be to go to the package insert, right? Yeah, multiple steps, including DNA digestion, precipitation chromatography, or tangential flow filtration. So I don't think much would get through, but if it did, it would be DNA encoding spike protein. And they're small pieces, most likely, because they've been digested with DNase, right? So they're small pieces. They're not going to code for anything. So it doesn't worry me at all. But, the press says, oh, there's DNA in the mRNA vaccines. And then they get a scientist to say DNA is bad because it can integrate into your DNA, which is true. It can integrate into your DNA. Yeah. But I don't know that that's mostly an issue, right? Most of our cells don't live forever, so I don't think it's a problem. So I just think that they're picking up on these stories that are not issues. Yeah. Patricia says Robert Malone was working on DNA biologics in the lipid nanoparticle. He couldn't get it to work in his animal model. Karen says, didn't know the Nobel Prize was this week. Now I'm wondering what is the origin story, history of the prize, and how projects and people are chosen, and would also like elaborating about COVID spike protein. Okay, well, we did some interviews with Erling Norby, who was on the Nobel Committee for a while, and he's written some books where he has talked about the how the prize was established. It was from Alfred Nobel, who invented dynamite and had a lot of money, and decided to. His idea was to support young people at the beginning of their careers, but of course it got warped over time, and now it's for people who are not always so young and have made outstanding accomplishments. His idea was to drive the discovery process, not reward it. But now it's a reward, right? So the question is it useful? So I think in one sense it's useful. And here's the only way that I think it's useful. It brings attention, widespread attention to science, because the prizes are widely reported, right, by many news outlets, and a lot of people, hey, did you see who got the Nobel Prize? A lot of people like that. So I think it's good for that. However, it does, it makes people want to get them and compete for them, and I just don't like that. And they also sometimes highlight just a few people in a field that may be huge and have more contributors than you can reward, so it's a bit artificial. So the whole prize awarding thing I don't really like. So I've never put Microbe TV up for any podcasting prize. I don't think that you should do that. I think you should just be recognized on the merits of what you do. Anyway, thanks for that. That's a good question. Can you tell me the author for the book Fever? John Fuller. John Fuller. Yep. John Fuller. I don't have it here. I have it in the incubator. Yeah, you remember a great TWIV with the author of that book about selecting. So I'm going to bring it up for you. It is TWIV Erling Norby. TWIV 564. Oh, this is cute. This is when I visited Sweden. Yeah, it was in 2019 from the 16th Smögen Summer Symposium on Virology. So there's Erling. We're at the front of the room there. I think I have the same shirt that I have on tonight. Wow. You'd think I only have one shirt, right? And these are his books. Nobel Prizes in Life Sciences, Nature Surprises. And then there's a cool article here about polio and Nobel Prizes. Why polio didn't get a Nobel Prize. That was cool. Nobel Prizes in Life Sciences. Yeah, this is a good history if you want to learn. So someone asked about the history. There you go. That's a good one for that book. So Kang also wants some follow-up from the immune episode where they discussed people with a worm infection were protected from COVID-19. No, that's it. That's it for that for now until we hear some people doing more. I'm not aware of it, but it takes time for this to get out, right? Vincent should go back to Lex Friedman's podcast. Well, the only reason I was on his podcast to begin with was because you guys wrote in. You wrote in and said you should get him on. You should get Vincent on. And enough of you wrote in such that Lex took notice and he got me over there, and we talked for over 3 hours. But I don't think he would want me back again. I think he's heard enough. But although he could ask me, like, is Joe Rogan right when he does this or that? Yeah, but he doesn't want to out his friend, right? Okay, on lab leak, are there reasons why it's less likely that lab leaks to lead to pandemics than a wet market scenario? Yeah. It's much less likely. First of all, SARS-1 started in a wildlife market. It's a risk factor. So here we have a wildlife market, which is the epicenter of early on in the outbreak. So it's most likely to be that. Why would it come from a lab when the wet market is right there? So my understanding is this, that so you have lots of animals coming in and out of the market. So animals are rarely infected, you're going to increase the likelihood that you're going to bring an infected one in by having so many animals traffic the market and encounter people, right? So, yeah. And also, the number of samples they have in the labs are not anywhere near as much as the animals trafficking in and out of the market. And so, and also the samples brought in the lab are typically inactivated. But, the biggest reason is that nobody had this virus in their lab. So I don't know how you can postulate that it came from the lab if they never had anything close to it, right? Nothing close to SARS-CoV-2. And you may say, well, they're lying. No. If they had discovered a really different coronavirus, they would have published it, because that's what scientists do. They publish it. Yeah. Can I ask Daniel about medication to give dialysis patients testing positive? Okay, let's see. I'm going to bring up the show notes for tomorrow's Daniel Twiv. I'm going to write it in right here so I don't forget, because otherwise I'm going to forget. That's going to be 10:50. And so Patricia wants to know. Patricia writes, yeah, I'm writing you an email. What medication should be given to dialysis patients testing positive? Now, I'll tell you what he's going to say. He needs more medical history, right? Are you talking about antiviral medication? Okay, will do. My quantum mechanics professor was probably sixth in line for the Higgs boson Nobel. Many contribute, only 2 or 3 can get the prize. Tom Kibble. Yeah, that's tough. So why do you want to reward just... reward just a few? I don't understand. I'm reading other questions here. Recent BC study said that Paxlovid only reduces mortality amongst the most vulnerable patients. So that's on the question list for Daniel tomorrow, okay? We're going to ask him that, because we got that question. Actually, you sent it in, Will. You sent it in. I'm just realizing I pasted it in from you. I want him to answer that, because he really has looked at those studies very carefully, okay? The Richard Ernst Award is one heck of a prize. It is. Let's take a look at it. Richard C. Ernst Medal. Let's see who got it. So, no, that's not the right page. Sorry. So Richard Ernst, of course, helped develop the MRI. And now I would like to go to, where's the page? ETH Zurich. There it is. Cool. So here we go. So here's the Richard Ernst lecture. And the last person to get it in 2022 was this guy, Professor Vincent Racaniello. An inordinate fondness for viruses. And this is the guy here giving the talk. I don't like this jacket. The lapels are kind of big. Anyway, it's a cool ceremony. Yeah. Yeah, of course that's me, but it's a really nice ceremony, and they treat you well. And I have the nice gold medal, which is at the incubator. You can come see it if you visit. Karen. Hello, Karen. There's Karen, my studio assistant. Don't be afraid of silence. No, never be afraid of silence. It means someone's thinking, and I'm not afraid of silence. I'll never forget that. I interviewed, what was his name? I can't remember, but he was quite an elderly guy. And he was slow in getting answers out. So I would ask him a question. He would give a sentence, and then I waited. I waited 10 seconds, and then he would continue. He just needed to collect his thoughts. Wacław Szybowski. You know, when a Polish name, Waclaw, Wacław, I think that's how you say it, Wacław Szybowski. And someone said to me later, you were brilliant to give him time, right? Yeah, that's cool. So silence is fine. Silence is fine. I heard something briefly about Paxlovid losing some of its efficacy. No, it's not true. It is, some of the more recent studies are very, very small numbers and different populations. So Daniel partially addressed that last week, but we're going to go through it again tomorrow. Once autoimmunity starts, how long does pathology last? Well, it depends, right? It's the antibody that's driving it, right? If the antigen is you, then you can't get rid of the antigen. You have to get rid of the antibodies. So the best thing is to switch off the antibody production, which could be done by immunosuppression, for example, right? So you can't really get rid of the antigen because the antigen is typically you. And so you could shut off the immune response with an immunosuppressant. Nowadays you can do it with antibodies against B cells, right? Like rituximab, which is an anti-B cell antibody, and that will destroy the B cells that are making the antibodies, and then you don't have the autoimmunity anymore. I don't know what the time lag is between the treatment and the effect, though. Candida auris. So what is the news saying? What is the news saying? Species of fungus grows as yeast, yeah, like candidas do. Multi-drug resistance, global health threat. CDC issues a warning about a fungus called Candida auris. Deadly fungal infection spreading at an alarming rate, especially in people with weakened immune systems, right? It spreads in hospitals. Okay, so it's kind of like Candida. People with weakened immune systems. Candida albicans was a bellwether for the early AIDS cases, right? Lisa said, I recently had pneumonia and was newly diagnosed with COPD. I wonder if the lung damage would make it easier for viruses to get a hold, worse than normal people. I'd be guessing, but certainly damaged tissues could lead to that, but I don't know what the mechanism of that would be. Chronic obstructive pulmonary disease. Let's see. COPD and virus infection. These lung and COPD become susceptible to repeated airway mucosal infections. And those are diagnosed as exacerbations of COPD, right? So basically the idea is that infection is a comorbidity of COPD. Yes, so you are right, Lisa, and there are in fact articles all about that. If mRNA can get into cells inside lipid membranes, why do viruses need to make fancy spikes that only bind target proteins when they could just evolve lipid membranes for themselves? Well, yes, so I'm going to say that it has to do with specificity, right? Because the spike on a virus allows it to infect certain cells. And so in the evolution of viruses, the spike protein or whatever attaches to the receptor evolves to get it into a cell that's internally permissive for the virus to reproduce, right? So otherwise it's less efficient, because if the virus is getting into a lot of cells, just by fusing with the membrane, many of those infections won't work, because the virus will only reproduce in cells that have the right internal machinery, and not every cell works for every virus, whereas the mRNA should be translated in any cell it gets into. So for vaccination, you don't need to have that specificity. Okay, so that's why receptor specificity is so important for virus infections. I have the flu now. Should I take the flu vaccine in a couple of weeks? I don't know. If it's 3 months like COVID, then you're going to be out of the flu season, right? Let's see. Let's see who recommends anything on this. Is it okay to get a flu shot after being sick? So apparently, let's look at CDC recommendations. They're saying as soon as you feel better, you can get it. But that's what I said last week. And flu vaccine timing. All right, that's just timing. Why don't we ask Daniel? Let's ask Daniel once again. Going to put it on the Daniel thingy-me-bobber here. Twiv. Sorry for the, all the sides. It's okay. You guys can chat among yourselves. So the question is, how long after getting flu? How long after flu can you get flu vaccine? Okay. Perfect. Thank you, Marianne. We will ask Daniel tomorrow. Is SARS-CoV-2 the only virus that infects ACE2 receptor cells? No, it's not. SARS-1 binds ACE2, and at least one of the common cold coronaviruses bind ACE2. And MERS coronavirus binds something different, and the other common cold coronas also bind something different. Yeah. Okay, Jupiter Crash says, Dr. Griffin said to rapid test on day 2, but someone read that new variant takes 4 to 5 to test positive. If Daniel Griffin says day 2, do day 2. A few days. And then if you get negative, you repeat it 2 days later. So, these are anecdotal reports that it takes 4 to 5 days to test positive. I don't understand why that would be, right? You have to be very careful about that. I would still do it on day 2. Yes, I heard the TWIV discussion was great, but no discussion of all the immune cells. I know, Susan, we have this dichotomy. We have some people who like the deep dives. We have some people who don't. I have to please everyone. But I'm not sure we know, right, what the function of these changes in immune cells are. They could speculate. But as I said, I will see if I could slip it into immune. There should be one coming up soon. Okay. I tested COVID positive Sunday evening and also followed Daniel's plan, but despite having game plan, took doctor hours on hold with 3-letter pharmacy, then rest of day to fill an unexpected struggle. Hey, you got it the same day. It's better than nothing, right? Vincent, last tweet was talking about algae as bacteria infected with phages. Is it possible to lyse the cells and put the phages in plaques to do a plaque assay? The problem is that the bacteria that the phage infect don't grow outside of the algae, right? So can't do that unless you can figure out how. But they're these are endosymbionts. They have very reduced genomes. They've lost a lot of genes that are needed for them to grow autonomously. So it's going to be very difficult to do that. Yeah. Okie dokie. Okay, it was Weissman and Cadico who won the breakthrough prize. Thank you. And Vanity says, very few passengers wearing masks on my flights anymore. Hygiene is also very poor. Yeah, it's too bad. But people are back to their old ways. They want to live that way. Yeah. Can you remind us how the immune cascade works? I think there's a mnemonic I've forgotten. I don't know which one you're talking about. You mean never let monkeys eat bananas? Neutrophils? The relative amount of each white blood cell. I don't know what you mean by mnemonic. How are the white blood cells classified? Maybe never let monkeys eat bananas. Highest amounts neutrophil, followed by lymphocytes, monocytes, eosinophils, basophils. Lowest amounts. That's the mnemonic I found. That's pretty funny. Pete's going to abroad. Yep. If you travel, that's not a bad idea. Okay. This was asked last week. Molnupiravir inducing mutations in RNA virus genomes. Is there anything to worry about? Okay. So last week I said no. It's just mutations on top of everything else that has been going on. Okay. And then I got excoriated for that. Do you know what excoriated means? Molnupiravir. And so I'm going to bring you the excoriating email from a molecular evolutionist. He says, I too patly dismissed molnupiravir's mutagenicity. Yes, of course, SARS-CoV-2 mutates. And yes, the spread of a given does not imply that it boosts fitness. But all that said, you should have been more clearly thinking for your listeners. 1, all else equal, widespread use of any incompletely effective mutagenic drug significantly raises the mutation rate of a population. 2, by definition, any strain that survived to get detected in surveillance descends from a virion that resisted the mutagen, any drug to which it was exposed. 3, nonetheless, a typical mutation, whether from a mutagenic drug or otherwise, likelier harms than helps fitness, whether to the drug or broadly. 4, but the more variation in the viral population, including by new mutations caused by mutagenic drugs, the more likely that some strains will indeed prove fitter in the environment than did the ancestor. As such, if we can choose between equally ineffective mutagenic versus non-mutagenic drugs, the latter may be the more sensible choice. And he said I should have provided more nuance. Well, I mean, these are all speculations. They make sense, but none of them have happened yet, right? And I don't know that they will. But I still see no harm if these changes are minimal and are not leading to any fitness accumulations. So I don't think it's a problem, Visto. I think the problem is that molnupiravir is 30% effective at reducing progression to hospitalization, and that's the problem. I got a tab shortcut on my bar that goes directly to the YouTube channel. Thank you, Questella. Appreciate that. That's what everyone should do. So Rach likes my idea. That's interesting. Approximately 98% vaccinated here. We've had an outbreak and 13 hospitalizations in the last few weeks here. I'll see what I can find out. Only people allowed it here are 70 plus. Oh, yeah, this Angela would be great on office hours. Yeah. And Angela. Yeah. And so we have a couple of weeks of no office hours, but that's a great suggestion. Yeah. Previously, you mentioned you could get tested for exposure to the capsid if you're vaccinated. So nucleocapsid, right? It's an internal protein. It's inside of the capsid. It's attached to the RNA. You could get tested for antibodies to that. That would mean that you were infected at some point, all right? That you got infected and the virus reproduced enough for you to make antibodies to the nucleocapsid protein. So what would be the reason? Now, just to know if you're infected, if you want to know that. Oh, I should show you the question I'm pondering here. Is there any reason why flu and RSV vaccines are not being taken on the same day? I don't know. According to CDC, flu, COVID, and RSV can all be given at the same time to eligible individuals. Okay? So there you go. Karthik. Last year, my wife. So I don't know how I pronounced it correctly. It's either Karthik or Kartik. So Kartik, I know because there's someone, a virologist at Einstein by the name of Kartik. My wife was infected with SARS-CoV-2. Since her recovery, she noticed a change in her menstrual cycle. Any known correlations? So someone asked this of Daniel, and he said yes. There are reports of changes in menstrual cycles after COVID. They're, did he say they were rare? Let's see. You're saying a scantier period, but there's also temporal changes. And there's a paper in Vaccine which says it reverses. The people who are vaccinated have a regular menstrual cycle. Yeah, there are a number of studies that have looked at that. And, Daniel said they were rare. But I'm not sure they're so rare. But I do think we have no idea what's causing them, Kartik. It's a good question. Vincent, can you remind us what caused Mickey did again? She was a guest on TWIV. Really? I don't think so. I don't recognize the name. Cause Mickey. I think the name is not right. Yeah, make sure you have the name right. It doesn't ring a bell to me. It's very disappointing that healthcare settings don't require masks. That makes it hard for the immunocompromised to better protect themselves. I agree. I think there are lots of patients in healthcare settings that have issues and would benefit by being protected. Now, they should themselves wear masks, but I think it's not a bad idea. But, Emin, here's the question. If we hadn't had a pandemic, would this ever have come up? Do you think even pre-pandemic, it probably was a good idea to protect them, right? because they are particularly vulnerable. Yeah. Vista loves the spike design. Yes, a lot of people like it, even though they don't know what it is. And while no one is wearing masks here in China, they have always been compulsory in hospitals. 2 weeks in the hospital in June confirmed this. Hope you're okay. And I had said last time I would go for an hour until the questions were exhausted. But you guys are always asking questions. It's great. I really like it. Don't stop. Okay? It's really good. It's true that the vaccine can cause menstrual irregularity and even cause it in some women. That's absolutely right. Yeah, I was just seeing that in some of the papers. Oh, Kiezmychia. Okay. Sorry, Tona. Kiezmychia. Not Kosmicki. Kiezmychia Corbett. Yes, the lady who worked on COVID vaccines at the NIH. Yes. So, yeah, she's just started in her lab at Harvard. We'll give her some time to get going and publish some papers, okay? Can point out it's a head-on representation and it's a trimer, 3 identical parts. Yeah, that's a good way to look at it too. The spike, you're looking at it down from the top. You're looking at the 3 protein subunits. Here's Karen's view. This is a spike protein, and it's found in COVID and the basis of vaccination. So it's not found in COVID. It's found in SARS-CoV-2, the virus that causes COVID, and is the basis of vaccination. That's a good one, though. Okay, this is a spike protein. It's found in the virus that causes COVID, and it's the basis of vaccination. Yep. So the spike T-shirt is the spike protein antigen molecule. Is this something that is part of the virus or something that our bodies create as a result of the vaccine? It's part of the virus, right? Yeah. Here, let me get you a SARS-CoV-2 virus, 3D printed by Mark Martin. And going over, I got a cramp in my foot. So these little protrusions, this is the SARS-CoV-2 virus particle. These little protrusions are the spike proteins. And they are sticking out of the membrane of the virus, and they allow the virus to attach to cells. Okay? And our bodies don't create them, no. And so you can take an mRNA that codes for the spike protein and put it in a lipid nanoparticle, and that's the vaccine. Or you can make the spike protein and inject it into people. And that's the vaccine as well. So that's what the spike protein is. I have to carry this with me everywhere so I can explain it to people. Your take on molnupiravir paper left me feeling I missed something. So I talked about it. It's error-prone, right. And my original argument was there's no, it's in a background of virus mutation, and the evolutionary biologist said I was missing the nuance. Because you just shouldn't use something that's not that effective. And that is true, actually. You shouldn't use antivirals that are not 100% effective at inhibiting viral reproduction. Because that is a recipe for mutation and resistance. Lisa, back to COPD. I'm still coughing from my pneumonia and recently discovered COPD. I wear a mask awaiting the newer shot. Wonder if COPD means masking forever. It's a good question, Lisa. It depends how often you're going to get infected and how it bothers you, right? Because if you don't mask, you're going to get infected. Even if you are boosted, you're still going to get infected, and that may trigger your COPD. Yeah, it could be a problem. Molnupiravir increases per sequence diversity in the surviving population, which might provide a broader substrate for selection to act on. So that's the point. it's increasing diversity in the population. I want to know what fraction of all the genomes out there have signatures of molnupiravir. And if it's minuscule, then it's not an issue because they're not fit. Yep. I work in a hospital and masking is sporadic. We've been told to wear masks if symptomatic. I also wear a mask when asked to by my patients. Masking is a difficult problem, right? I think in a healthcare setting, all people should mask most likely. Yeah. Dr. Koopman shared a study in which vaccine mRNA was found in ventricles of only 3 patients at approximately 2 copies per nanogram. Besides being few patients, what is this measurement in relative terms? It's not a lot, right? So a nanogram is a good amount of RNA. I don't know how to put it to you, though. It's a millionth of a gram. It is very, very low. How much? Let's try and see how many nanograms of RNA in a cell. Okay, so a cell has about 0.025 nanograms. So it's less than 1 copy per cell. 0.025 is the number, total cell number. So it's vanishingly small and probably not even intact. Okay, so Bhutan has one of the best vaccine coverage and strategies during COVID. I wonder what that would be. That would be interesting to know about. Do you think any future new vaccines will be inactivated, or is that method obsolete? I think it's gone. I think, there were a couple of inactivated COVID vaccines, right? They didn't work so well. I think vectors and mRNA technology are superseding it. Yeah. It's a good question. I do think, it's gone. There was a Nobel Prize for using malaria to treat syphilis in 1927. How did that work out? Can you explain in simple terms the theory of how mRNA technology can vaccinate against cancer? So the idea is that cancer cells have a protein that's not present in other cells. We call those neoantigens. And one of the ways that they arise is that cancer cells are dividing continuously, so they're mutating. And they're making some new proteins that aren't present in normal cells. So if you can identify those, then you immunize against them using mRNA vaccines, then the cell that your body will attack the cancer cells and kill them. Yeah, that's the idea. So back to the molnupiravir mutation rate. So you ask me if I'm worried. I say no. And then I explain why I'm not worried. It increases the solution space for selection. Maybe. It depends on what fraction of all the mutations out there are those, right? If they're vanishingly small, then I don't think it's significant. The data Lori's talking about is not yet published about females and autoantibodies in long COVID. Okay, well, all I know is that the Nature paper that we covered on TWIV, they didn't find any correlation between autoantibodies and long COVID. So I don't understand why you'd find something else in a different study. You make a big paper saying there's no correlation, then you have another one saying there is. So I have to read the other one. I don't get it. Yeah. Does anyone else listen to the podcast Nobel Prize Conversations? The head of the Nobel Prize making the phone calls to the recipients, and then they have a short conversation about their work. Oh, that sounds cool. We are happy to tell you that you've won the Nobel Prize. Is this a joke? That's cool. Why can't Amy be on the live stream? Because she is a government employee and they don't want her to do it. That's basically it. So I haven't banished her. She can't do it anymore. In fact, I was talking with her tonight, just before, and I said, she said to me, are you looking forward to the live stream? I said, hey, why don't you join me? She said, no, I can't. So I tried. MN has 2 Spike shirts, a hoodie and a coffee mug. Very cool. Very cool. I've been planning to rewatch the virology course, but wanted to wait long enough so my brain goes through a sort of somatic hypermutation before the boost. Totally. Why don't you wait till the spring when I teach it again? It's going to start in January. You know, I'm going to make some changes to it. I want to try some new stuff. Yeah, that's the idea. But whether I'll have time to do it or not, I don't know. But yeah, wait till then. Okay, so now it's 3 people have asked about Amy. Okay, Amy, if you're listening, 3 people have just asked about you. H. Yes, H was that. It was a name I gave. Is TWIP always going to be a question answering format? I don't know the answer to that. It's worked for a long time, but I have a feeling we're losing our audience. You know, the numbers slowly decline. I mean, tonight we have 190 people. That's not bad. Usually we have around 200. Oh, by the way, hit the like button. We have 169 likes. I think some of these podcasts have to have a reboot. You know, we did a reboot on TWIV years ago to go to the question format, and it worked well. But you know, I feel that when we're reading the questions, sometimes it just goes on, and people lose interest. So I have to talk with my co-hosts about that, about what to do. Oh, and Sharing enjoys listening while working in the biosafety. Thank you very much for listening. Next time I'll say hello to you. I'm going to write it down. Say hello. Or I should shout out. TWIV. Shout to TSHERING. Just first names. That way you can, oh, it's me. That's funny. I love those Vincent promo photos. To get here, I typed TWIV live and got many live TWIV, not live streams. Well, Karen, why don't you subscribe to the YouTube channel and then turn notifications on? You'll get a notification from YouTube when it's ready to go. What about SV40 detection? So, Dean, I mean, look at this. I know what you're talking about. So the people who claim there's DNA in the mRNA vaccines, they say there's SV40 promoter DNA in there. So that may be the promoter that's used to do the... No, it's not. Sorry. It's T7 RNA promoter. So I don't know what SV40 is doing in there. SV40. If there is any, it's just vanishingly small and it's pieces of nothing. Let's see what they're saying. Claim that COVID vaccine contains DNA. And then there's another rebut, saying no monkey virus DNA was found in COVID vaccines. Primate. It's not there. It's wrong. So, no, there's no SV40 there. You think I have the flu? I've been sick for 8 days. Well, that's kind of long for flu. Flu is usually shorter than that. Unless you're having a post-acute sequelae. Many people develop pneumonia after the first flu course is over. So you might be careful. You should be careful. Plenty of other things could contaminate anything being manufactured. Yeah, but they are checked, right? Quality control. I don't think small pieces of DNA are a risk. If you think it's a bigger sign of contamination, that's another issue. But then it's out of my league. I don't do quality control. Not to imply that I think there's a big risk. These are GMP facilities for a reason. Exactly. Thank you very much. Patricia, Dixon is a national treasure. He's my dad's age. Almost lost my dad last night due to cardiac arrest. He's fine now. Had 3 stents. Never know how much longer we have with our elderly. Indeed, Patricia. On TWIV and TWIP, you could put another window in the middle to show us the figures. Yeah, we could. David, who is my video editor, tries to do that. But, there's how much time can you put into editing, right? And the cost goes up because he's paid hourly, and you have diminishing returns. How much is it appreciated, right? I just wish it were easier to put that in. But I think it would be good. I agree. Yeah, that would require a lot of editing. And then we have me, Dixon, Daniel, and Christina, 4. So we have 4, which make a nice square, but then you put one in the middle. It's going to ruin it. Excuse me. Sorry. It's going to ruin it. Nobel was reported to have died, and newspapers ran as a bit, which he read. He wasn't happy with what it said, so he decided to establish the prizes. I didn't hear that one, Craig. He did have a tragic life, okay. What are the current variants of concern? Sorry. Let's take a look. Let's see if we can find some. Covariants. Let's see if I get the right. Covariants.org. So let's share this. Okay. This is covariants.org. There you go. And here are all the colors, right? So let's see. Per country, do we want to see that? Nah. these are so crazy. So here is April. This must be where we are right now, right? The end of 2023. And I don't know what color this is. Oh, this is what? 23F, Omicron derivative. So you see we have a whole bunch here in the U.S. So if you go down at this time point, these are all the variants F, B, D, A. They're all Omicron derived. So they're all playing around, and my understanding is that these are all neutralized with antibodies induced by XBB.1.5. So nothing is jumping out at me. No. Yes, the polio Nobel went to Enders for growing virus. He developed a way of growing it in cells in culture, right? Not for actually making the vaccine. Ian, thank you so much for your support of science communication. I did get your email. Thank you very much. I will follow up on it. Vincent, I discovered Lex on account of you being on his show rather than vice versa. It's funny. Yeah, Lex would not think that's so good. My husband and I never got COVID that we know of, got all our vaccinations. Can you test to see if you had COVID without being sick? Yeah, you could. It's something we mentioned earlier tonight. You could look for antibodies to the N protein. So you have to get an antibody test, and then you could ask if you were ever infected. Yep. The Chinese hospitals would ask the people with the new pneumonia-like symptoms basic background questions like where they worked and the market and related market kept coming up before any hypothesis. These were certainly, this is the early epicenter. Most of the first patients had some connection with the market. Not everyone, but you wouldn't expect everyone to, right? Because if someone went to the market and got infected, then they went and infected someone else. That other person wouldn't have any connection to the market. Especially as time goes on, you'd have more and more of those, right? I think the claim that the coronavirus lab leaks, which have happened, is dicey. What is a real documentation, citation of any corona lab leak? There is none. There's no evidence for it at all, and people say, well, they're hiding it, but that's nonsense, okay? There's no evidence. We have plenty of other evidence that it is not a lab leak, so... If he wrote that, which have happened, I agree, Tom, that I missed that, and that's not correct. Scientists may be constrained in what they can share publicly by perceived or real national security considerations. Well, not a scientist like me. I'm not doing anything that can't be shared. Nobody tells me what to share or not, or told me what to share or not. So, I don't know. Maybe in China it's different, but if you're working on coronavirus, nothing's not shareable in the U.S. as far as I know. John says, I seem to remember a French horn. Yeah, you're absolutely right. So they played a French horn quartet, which was called CD 155. So it was written in my honor. And the first, so CD 155 is the name of the poliovirus receptor, which was discovered in my lab. And the first 5 notes of the piece were CD 155. It's very cool. I'd love to get a recording of it and play it somewhere. Yeah. Yeah. Dixon's son was part of the jazz quartet that played at TWIV 1000, right? Okay, so I saw this question a long time ago. Vincent, I can't believe more people aren't interested in this live stream. Maybe send out notices to PhD programs or med schools, something like that. Well, I think we have to advertise it more, right? I mean, a tweet goes up, and an Instagram goes up a couple of hours before. It's not enough. I totally agree with that. I need a social media person to blast our stuff out there. I need somebody. So if you know someone who would like a part-time work doing good social media for a science communication company or a science education company, where every day we have 4 or 5 blasts on Twitter, Instagram, Facebook, LinkedIn of stuff that we're doing, clips from what we're doing, advertising the live streams multiple times way in advance. Just someone who knows how to leverage this. I would love to hear from it. Because, yeah, I think we should have more than 200 people. Not because I need more than you guys. You guys are good for me, but I can't ever even answer all your questions, right? But just so people can learn, because a lot of people who come don't ask questions, and that's great. They learn by absorbing. So if I had 900 people, 1,000, we used to get 1,000 people, but a lot of people had COVID questions, but they're all learning, and then they're going to pass it on. So help me get publicity. Would anti-B cell treatment for autoimmune disease have to be lifelong? I don't know. That's a clinician question. I don't know if any of them ever resolve, or it's lifelong. I don't know. Should I ask Daniel? He's not really he wouldn't be doing that. Let's see. Is autoimmunity a lifelong disease? Yeah, some require lifelong treatment to ease symptoms, according to the National Institutes of Health. Yep. Is hydroxychloroquine effective and useful as an alternative? No. Hydroxychloroquine doesn't actually inhibit virus reproduction in the lung. So it's not effective at treating disease at all. Paxlovid is much better, yeah. Lindsay, you're the GOAT. Don't need anyone else, maybe a few guests now and then. You cover so much in 2 hours, help so many people. No one else does Q&A live from chat. My 2 cents. Thank you. I forgot your first name. I'm going to look it up because I have it in my email, because I feel silly that I can't remember. Now my stinking Outlook went to the new version, which I hate. I hate it. Ferraro. I know you sent me a couple of emails. And the new one doesn't search quickly. Look at this. This is so stupid. Well, while that's looking, thank you very much. I appreciate it. But people like a little bit of diversity. I don't mind having guests in. Did Daniel Griffin say to test in the morning? I think he would have sent it to Vincent at microbe.tv. It's not even showing up. Oh my God. I want to go back to the old one. Revert to legacy. No local mailboxes. I keep telling Microsoft I don't want the modern one because you don't have any local mailboxes. Okay, did you send it to Twiv? That could be. Ferraro. Here it is. Gwen. Monthly memberships at YouTube. I applied for it. I haven't heard from them. Did Daniel Griffin say to test in the morning? I don't know, but I don't think that would make much of a difference, right? But I mean, it's a good time to do it because before you go out, you could see if you're positive, and if you are, you don't go out, right? Is it true that some viruses cause cancer? Yes. A handful of viruses do cause cancer. It accounts for I think about 22% of all cancers are caused by viruses, like human papillomaviruses, Epstein-Barr virus, HIV even, hepatitis B and C viruses. Are viruses good for anything positive for humanity and life on Earth? And if so, what? So they're really good for life on Earth. They're really important for turnover of organic materials. So all the cycles, the carbon, the sulfur, the nitrogen, the phosphorus cycles, they all depend on viruses degrading organisms in the ocean. They move genes around, which is really important. And we think they're beneficial in other ways. For example, in mice at least, they seem to help development of the gut in the case that bacteria are missing. So I do think that they have their uses. So it's harder to show experimentally in people that viruses are beneficial, but you can show it in animals, you can show it in plants. They make plants thermotolerant, for example. They make them drought tolerant. So I think they get a bad rap for obvious reasons, right? So I think there's more good than bad. Yeah. Any good chance of new antivirals for more people for COVID? Yeah, I think so. I think people are working on it. I think it's going to be some time before we see them. But, there's still a lot of infections globally. So I think we're going to see them. Intranasal vaccines, people are working on it. I don't know what the key is because the flu intranasal vaccine isn't really any better than the intramuscular vaccine. So people are going to have to come up with some good ideas for improving them, right? So I'm positive. I think people are working on it and I wish them well, but I can't tell whether it's going to succeed or not. Audience, John wants to know, what should I watch? I don't know what people have said. Let's see. If someone takes time to develop a question, it's okay to take time to answer it. Oh, yeah, I understand. I don't mind taking time. I don't want to go on for an hour per question, right? That's not the point of a live stream. I think you do have to have a limit on how much you answer. And so I try to give thoughtful answers and balance it because we have a lot of questions to answer and can't go on with them forever. I would do my best. Twin is my favorite. I like Twin. They're very, very interesting topics, especially the latest Twin is all about how opioid addictions are learned, environmental learning. It's very cool. Oh, for the immune response, the IgM followed by the IgG. That's very simple, but that's probably not what you meant. Question for Daniel. Is Novavax vaccine still worth getting, or has the spike mutated so much? I don't know what the status of Novavax is. I don't think that they have ever made enough. And, they were going to adjust it. Yeah, it's been updated to include XBB. So if you want to do that, I don't think it's so far behind any longer. No. I was wondering if you're mainly a reader or do you sometimes listen to audiobooks? I don't. I don't because I read in the train when I commute. Yeah, I don't listen to audiobooks. Sorry about that. 9:54. I bet everyone's chomping at the bit to leave. Let's see what else is left here. Where's the shirt? Vaccinated.us. The spike shirt is right there at vaccinated.us. Yep. Yeah, cancer and transplant patients are immunocompromised and they should be protected in healthcare settings. I agree. Hello, Carol, our favorite nephrologist. Nice weather. A nanogram is a billionth of a gram. Sorry, yes. Not a millionth. Sorry. SRR, thank you. Oh, where did it go? Here we go. SRR, thank you for your contribution to science communication. Michael Min is complaining about Paxlovid rebound. I don't understand why people think it's a thing, because it happens without Paxlovid. So how do you understand that? How many nanogram of spike would be found per cell if one caught the infection? That's a good question. Nanogram spike per infected cell. I doubt I'm going to get it by Googling it. Here we go. Each infected person, blah, blah, blah. No, that's infected. There was actually a wonderful paper a while ago that it estimated the total number and mass of SARS-CoV-2 virus particles. So every person has a mass of 1 microgram to 100 micrograms. So, the nanogram of spike would be much less, but I can't give you the number, and I don't want to do it now, but I bet we could figure that out. That would be interesting. Will the incubator be open to visitors during the Christmas holidays? Yep. It will be open. Just email me, vincent@microbe.tv, and you can arrange to visit. Yeah. I read somewhere you were going to Milan. Yeah, I already went. We went for 3 nights, and it was wonderful. It was really a nice trip, but the meeting was good. I didn't do anything else. For the dengue vaccine, how about those 3' UTR mutated attenuated vaccines by Whitehead? Yes, those are very interesting, and they're working very well. I like them. Oh, Peter likes TWIP with guests who suggested the case. Yeah, that worked well last time. Maybe we should do that more. That was a really cool case, right? See, 4 or 5 people are now saying, very sad, Amy can't be on. Oh, notifications from the Discord work very well, too. My YouTube notifications work well enough, but they're famous for not working from time to time. So if you sign up for Discord, you get notifications of our live streams. And you have to go to one of our programs to find the Discord link. Should we get an Amy puppet? No, we can't get an Amy puppet. Here is the... to stand in, but we can get Amy the virologist. There you go. So I could have her right next to me here, okay? Or I could have her in the back. How's that? There you go. Can I just share screen? Oh, yeah, for the shows. So I'm unfortunately the only one who can share the screen on the software that I use. So I could do that. But, I have so many things that are going on on a recording that it's not easy. I need to have a producer in the studio. That would be great. Vincent, from First Principles, what do cells do if random segments of DNA are found in the cytoplasm? Well, the cells get rid of it because it's not supposed to be there, and they're sensing mechanisms for getting rid of DNA in the cytoplasm. Yep. Okay, Vincent. Oh, I just answered it. Yeah, there are sensors in the cytoplasm that get rid of the DNA. Okay, let's wrap this up. Let's thank Lisa. Thank you so much for your support of science communication. Silvio Pina, thank you for your support for science communication. Don't forget, you were just telling us about the herb that created the placenta. Yes, so there you go. Viruses made that happen, made us have placentas. Thank you for reminding me. And there are other examples of how endogenous retroviruses and their gene products have helped us. Look at all the Miss Amy's here. So now I can tell her that a lot of people missed her. All right, folks, thanks for coming here tonight. 200 people. Yeah, wonderful. Love you all, baby. And I want to thank our moderators tonight. We had Tom, we had Les, we had Vanity Nutrition, we had Steph SF, and I think that was it for tonight. Thanks to everyone for coming and bringing your wonderful questions. Really good. And as far as next show, so next week, I am supposed to be in Boston, so probably, and no show there. And then the following week in Chicago, so no show there. So the next show is going to be, let's see, hang on, folks. It's the following week, but I want to just get it on the calendar. That is October 25. So it's quite a bit of time from now. Don't leave us. Make sure you come back in 2 weeks. And in the meantime, of course, everybody, I'd like you to be safe. I'm going to turn off Amy here and turn on our plaque and say thanks for coming, everybody. Good night.